systems level models today, and this is something that, as imagers, I think a lot of you are probably familiar with. I love to give this talk to people who aren't imagers to sort of show them that how powerful imaging can be in driving these models. But I think it can't hurt for you to see it too. So the question of why and when systems level models are important. So there's a general sort of concept in the literature, and there's a paper by Banks and Tran that talks about iterative modeling in biomedical science that gets at this issue of finding some sort of clue and then taking that, looking at the literature, existing literature, and starting to put pieces together to solve problems. And then once you put the pieces together, you can actually ask new questions and collect more data and get more clues. And so basically, it's this process that goes around, and you just keep going around. And each time, you know a little more than you did last time. So you might start out with a systems level biomarker, such as you would find in imaging, and then you start searching the literature while you're looking for ways to explain your finding, not only at the systems level, but say at the level of pharmacology and anatomy. And then you tie it in with ideas about that you already know about functional systems, like the motor system, for example. And then you can generate new hypotheses to test based on the pieces of information you put together, such as, for example, using a biomarker to subtype disorders. And then basically you come up with new ideas, you test them, you get new data, and you start the process all over again. And each moment you get closer and closer to building a nice model. Now, there's some challenges to putting together systems level models, and in particular, when you have a complex system, and pretty much all systems in the brain are complex. So often we don't have the information at the neural level to be able to build a model that would one-to-one match what the brain is doing. In addition, a lot of the models that are out there that people put together aren't amenable to even bringing down to the neuronal level if you tried. So it's really important. With a background in basic neuroscience, I feel very strongly about being able to tie a systems level model down to the level of pharmacology, anatomy, single unit recording. It needs to be consistent with all of that data at the lower levels. So many models only explain one aspect of a functional system. For example, they might model healthy function in a system, but they aren't able to model disordered function of the system, or vice versa. So one of the challenges is try to build a model that can explain both. Clinical data is absolutely critical in putting together a model because that gives us a lot of clues, just like imaging does. But the tricky thing about it is it often doesn't match one-to-one with mechanism, and that can be really misleading sometimes. You say, okay, we're going to correlate our findings with this symptom. It doesn't always get you back down to the mechanism. Models often also don't cross-reference other fields of research. In putting this together, I've looked at a range of research from kinematics to molecular biology to computational modeling. It really helps to read across literatures. And I know a lot of people do that, but I know when I go to conferences, I often see these people who, especially with imaging, they're just talking about imaging literature. So I feel very strongly about tying all of these other literatures in. And often, actually, I discovered there are some insights in other sets of literature that weren't in the field of neurology, but other people had actually discussed ideas in kinematics that had not sort of made it over to neurology. In terms of mechanism in models, quite often there's a discussion of that the system is disorganized, and you get this disorder because the system's not functioning properly. My question is, if symptoms are stereotyped, if you can define something as a symptom, there's something about it that makes us call it that symptom. And later I'll be talking about the movement disorder dystonia, which is very stereotyped and is a perfect example of this. So why would something stereotyped result from disorganization? An alternate possibility is that in some cases the symptoms actually reflect an amplification or downregulation of a discrete functional system that is normally integrated so smoothly with the other systems that you don't necessarily recognize it as something that you would see as normal function. So studying disorders can actually be extremely helpful if we can say, well, this disorder an amplification of the movement system, the postural system. In other cases, in mood disorders, which I study as well, is that an amplification of, for example, do you have changes in the mood system where you upregulate or downregulate reward or aversion function? So to give you some examples, we know how complex these systems are. We already have some models. So this is a basic sort of circuitry diagram for motor control and circuitry diagram for motivation and emotion. So we already have a lot going here. So there's a lot to build on, which makes our job easier. So let me just first sort of describe the current dominant model for motor control. In the field of neurology, at least, and I'm sure in each field it gets a slightly different take. But this is sort of basal ganglia centric, and the most common model that you see, and there are different variations of it, but what you most commonly see is there's so with the basic basal ganglia circuitry, you have the striatum, you have like the putamen, and that basically projects to the globus pallidus. And in between somewhere there you have the subthalamic nucleus. But the exact anatomy isn't important here. The point is that we know there's certain circuitry there, and there are two different main sets of pathways that project in slightly different ways. Classically, one of them is called the indirect, one of them is called the direct pathway because it goes straight to the internal part of this structure, the pallidum. The other is the indirect pathway that has a stop over at the subthalamic nucleus before getting there. So classically in the literature, the direct pathway is seen as selecting motor programs. And basically that then goes up to thalamus and cortex and disinhibits the motor programs that you want to select. The indirect pathway is thought to inhibit unwanted motor programs, meaning that every time you make a movement, you also have to inhibit all the movements that you don't want to make. Which makes sense. I mean, you do need a good mechanism for selecting movements. But I'm going to argue later that that's not a very efficient use of circuitry. So what are the implications of this classic model for movement disorders? Well, this is from a paper by John Mink in 2003, which is sort of, again, it's this dominant model. And it shows these diagrams of basically a center surround, arguing that the selection of movement is based on a center surround mechanism similar to what you see in the visual system. And that, for example, that you're going to select this movement here and you're going to inhibit all the ones around it. And in certain disorders like dystonia, you have a change in the amount of inhibition. This should all be fully inhibited, but you have less inhibition here. And I'll explain a little more about dystonia later so you understand more about that. In other disorders such as chorea, you have reduced inhibition as well, but it's a slightly different pattern of reduced inhibition, and it's sort of more global. And in tics, for example, you would also have more focal disinhibitions of unwanted movements. So this is all movement-centric, arguing that the motor control system is just, basically there's movement and there's inhibition of movement. So how can we improve on this model? Now, the first thing to say is that it's really important to have this model and to have all of that circuitry mapped out. Without that, we couldn't build an even better model. So the first problem with this, if we go back here, all the explanations for different disorders are simply quantitative, or where is the disinhibition happening? There's nothing here that really provides an argument for why different movement disorders look qualitatively different. Why does dystonia look different from chorea? Why do people with Parkinson's disease look different from people with tics? It seems like a very simple explanation just to say, well, it's in a different part of the basal ganglia. So another thing that I think is important is that if you know anything about the basal ganglia circuitry, the indirect pathway is actually so complex. It has so many connections, and in fact, the more research is done, it's pretty clear that those two pathways aren't distinct. They run in parallel, but everything is pretty much feeds back to everything else. To me, it seems surprising that in nature there would be such a complex set of circuitry, simply to it's sort of redundant to the motor one. Basically, in this here, it's basically, so the direct pathway is doing the job it needs to do, and then the indirect pathway comes in and sort of gives backup to provide a little more inhibition of the system. I believe that this circuitry must have its own role. Especially given how complex movement is, it would make sense to have two parallel systems that are actually doing slightly different functions, but both contributing to motor control. So we're going to discuss how we can do that later. So then there's the problem with center surround. Center surround works great for a finite representation like on the fovea of the retina. It's a little harder to actually map out when you are doing something like storing programs. There's a difference between a 2D representation on the back of your eye versus storing programs that you use all the time. If you actually try to map this out, it becomes very complicated because you either have to have each neuron, each program has to reach out to all the others and suppress everything around it, so they all have to be connected all the time. Or you have to have, for every representation of a motor program, around that motor program you have to represent every single other motor program that's stored somewhere else. So if you try to actually draw it out and make a model, it's very difficult. So, and finally, in movement disorders, we know especially in things like dystonia, we know that there are a lot of different things that can cause the same disorder. So you get a stroke in the thalamus, you might get dystonia. You get a stroke in the putamen, you might get dystonia. Patients with Parkinson's disease, they can get dystonic symptoms both at the peak and at the end of their dose of L-DOPA, meaning that you can get dystonia both with too much dopamine in the system and too little dopamine. So how do you build a model that can account for all of these? So what role does imaging play in this process? And since I'm talking to an imaging audience, I wanted to make sure that this was an imaging-centric talk. So the clues that we get from imaging, you can often have just one finding that when you start searching the literature and tying things together, it gives you this sort of moment of, oh wow, I wonder if this is what this does. In this particular case, there's some literature in the anatomy of the basal ganglia that the authors actually said, it was some beautiful histologies, beautiful track tracing studies, and they actually said, you know, we wonder, there are two different subtypes of neurons that project from the internal pallidum, so the basal ganglia. The question is, what are those, what is the purpose of that? Why are there two different types of neurons? And so at the systems level, you can start to answer questions like that, or at least develop hypotheses. So I'm going to sort of split the talk into two parts, and this model is actually pretty complex, so and there are a lot of details, so please stop me if there's anything that I'm not clear about, and hopefully I'll have enough time to get through everything. But if not, I think there will still be plenty here. So the first thing I'm going to do is explore how clues from imaging itself generated some new ideas about the functional architecture of the brain, and then move on to how new ideas about dystonia itself can tie back into this model and be further tested with imaging. So this is getting at the iterative process here. So first, clues from imaging. So I'm going to start, our model for imaging is a movement disorder called dystonia. And dystonia basically causes involuntary muscle contractions. It can be anywhere from focal, like writer's cramp is technically a dystonia, to very generalized, so that it's affecting all parts of the body. And It does not have one cause. There are at least, I'd say, about 20 different potential genes that we know are associated with dystonia. So it is really a perfect example of a systems level disorder. So I'm going to show you an example of somebody who has a fairly generalized dystonia here, just so you can see what it looks like. And the arm is not part of the dystonia. He's trying to pull himself up. His trunk is spasming sideways, basically. So now that's not the most severe case I've ever seen, but basically it's this issue of the muscles are contracting involuntarily. And you get a lot of co-contraction, both of groups of agonist muscles and co-contraction of agonist and antagonist muscles at the same time. And this is actually what makes people think that in dystonia you get co-activation of competing movements, because you're getting muscles that are actually antagonistic to each other in movement happening at the same time. For example, I'll just give you an example. In the neck, moving the head this way requires certain muscles. But while you're doing that, there are other muscles you don't want to activate because they would actually counteract the movement, and those would be the ones on the opposite side of the head. So if you have co-contraction of these two muscles here, and the muscles on the other side are contracting at the same time, you're going to get this really stiff neck, and your head's going to start moving around. So in order to study this disorder, we are using a combination of a bunch of different imaging modalities, and the example I'm going to give you at the moment is diffusion tensor imaging, which you are probably all familiar with. So the important thing with the DTI signal that I'm going to emphasize is that not only that it measures water diffusion, and so I'll just give a summary. I'm pretty sure all of you probably know what this is, but in case not. So basically, it measures water diffusion, and the more the water is flowing only in one direction as opposed to many directions, as you would get between axons in bundles of axons in white matter, the higher the signal of anisotropy, basically. And the lower, conversely, the less it's going in one direction, and it's sort of flowing all over as it would in a cell body or between cells, the lower anisotropy you have, the more isotropy. So there are a few things that can affect this, and one, obviously, since originally DTI was used for stroke detection, if you have a loss of cells, you're going to have a lot more water diffusion in many directions, and it will also diffuse farther. But the way it's most commonly used to study disorders now, or healthy brains, is to look at the issues of myelination and potentially density of axons. Like somebody who's more skilled at something, do they have increased anisotropy or lower anisotropy? But the other thing that it's going to be important here is the issue of coherence. So if you have a bunch of axons, and I'm going to show you a diagram of that more later, but just remember the coherence thing is going to be important. So if you have axons that are all running straight together like you do in the internal capsule, that are all kind of projecting straight down, you're going to have much higher anisotropy. But if you have a bunch of axons that are one bundle crossing another, or if the axons themselves have collaterals in them, you're going to get a lot less anisotropy. So what we found when we put dystonia patients in and scanned them with the DTI sequence is that there was, so I'll start out by saying we were going to look at the basal ganglia circuitry to start with. So this is the area we were really interested in. This is our motor region. We know it's involved in a lot of movement disorders. So it's hard to see here on this colorized image, but this is the putamen here. The pallidum is here, and then this here, the red corresponds to is going in the x direction. So basically, there was an asymmetry in what looked like the fibers projecting out of the pallidum toward the thalamus. And you can see when you compare it to the control that you have a lot less, that this looks the same on the two sides, and this looks quite different. And and you also have a lot more of these fibers here. So this is the initial finding, and it's actually quite old now, that gave us this idea that I'm going to get to in a second. We also found, so this is more recent data, that we can basically replicate our finding around the asymmetry by looking also at tractography. So one of our questions with this, how do we know this is something that's related to the basal ganglia? There are a lot of fibers that run through this region, including things like amygdalofugal fibers, and the anterior commissure runs just anterior to that. So we used seed regions for tractography, where basically we looked at this is probabilistic tractography using the FSL software, and basically segmented both the pallidum itself and the region that corresponds to the one of the main output pathways called the axial lenticularis that runs where we have this finding. And we looked at the tractography from them in patients versus controls. And so what we found, so basically the blues are decreased in patients relative to controls, and the reds, yellows are increased. So what we found is in the left hemisphere, and these are cervical dystonia patients, so they had a neck dystonia, that there was decreased tractography from the left axial lenticularis. So the little bit of green here, that's the axial lenticularis. So we use this area as a seed region, and we have less projecting out of that region in the patients than the controls. And you can see here a 3D rendering of it, and it really, it's nice because you can see it actually coming out of the pallidum. So the seed region was actually in this area, but you can see that it actually goes back in there. So it's more convincing that this is actually a pallidal output pathway than with just the FA data. So, and then it projects down to the brainstem, and also looks like it projects up to the thalamus. On this side, so the right pallidum, we also saw differences, but it was an increase in patients relative to control. So again, we have this left-right difference. If this is not a relative left-right difference in patients, but it's a difference relative to controls. And in this case, the findings were not, we actually didn't see differences in the region here where the ANSA was. It was further back. So both of these factors here, both the location and the difference in direction, are consistent with the idea that what we saw previously really is a difference in the amount of output, or in the features of the fibers being output from the pallidal region. So what is so intriguing about this? Well, when you see something like this, you start asking what's going on. Is it that is there a fiber loss in this region, or are there actually more fibers on one side than the other? Is there an imbalance of fibers? There are a lot of different possibilities, but the basic idea is that it's not the same on the two sides. So while I was looking for, I was exploring both the issue of reduced numbers or increased numbers of fibers and issues of coherence. And while I was looking about searching ideas about the coherence of fibers in this region, I discovered a paper which shows that there are two major motor output types from the internal pallidum here. And this is one of those track tracing studies in monkeys that is so elegant. I have so much respect for people who can do this. So basically in one type, it's fairly simple. It projects out from the internal pallidum, and this is where it enters the thalamus. These are thalamic nuclei here, sensory motor nuclei, and also projects down to the brainstem. And this is actually something that in the basal ganglia literature, people tend to not talk a whole lot about the projection to the brainstem. It's mostly the thalamic projection that is looked at. However, the second type, you can see here, it's projecting to sort of the same areas, but it's projecting to a lot more areas. Not only is it going to the thalamus and the pedunculopontine nucleus in the brainstem, but it's also going to the red nucleus and some of the paraventricular nuclei of the thalamus as well. So this is projecting more extensively. But what is even more interesting here is look at the length. Look at the length of that axon versus that one. This is huge. This is centimeters. This is in the brain, 27 centimeters. So it is so convoluted. It projects so many different places, and it sprouts all of these collaterals to all these different places that it is actually 27 centimeters long, and this one's just under 6. So, and this was the paper where they basically said there must be some reason, some functional reason to have these two different types of neurons. And if I hadn't done this imaging, I never would have started, I wouldn't have found this paper. So the imaging is what led me to this. But the imaging didn't only lead me to this, it led me to identify the possibility that maybe there are different densities or numbers or configurations of these two fiber types in people who have dystonia. So it's identifying this paper, and it's also identifying its relation to dystonia, and that one or the other of these fibers may have some relation to what is going wrong in dystonia. So just as a schematic to sort of understand how this relates to the diffusion imaging, so this is just an example in in a healthy individual where you would have both of these two fiber types, that sort of the slightly collateralizing and the extensively collateralizing axons, and they're coming out of this internal pallidum here, and they're going both to the thalamus and to the brainstem. So obviously, in the case that you have only these ones that do not collateralize a lot, you have very high fractional anisotropy because they're all aligned. Once you start bringing in the collateral, by definition, if they're collateralizing, they're going off at some angle, so you have a much bigger reduction in FA. And you can see when I zoom into the area that's between the pallidum and the thalamus, this is sort of the simple type, and this is the highly collateralizing type, and you can see how many more directions this one axon goes in. So in dystonia, one possibility is that this asymmetry is resulting from more of these collateralizing fibers on one side than the other. Another possibility is that there's the same number of fibers, but they're collateralizing more extensively. So we obviously don't know which it is, but it sort of begins to tie in the idea that these fibers might have some important role in dystonia. And that's what I just said. So I'm going to sort of shift now a little bit, and then I'm going to tie things back with what I just showed you. So shift to this idea of stepping back and thinking about dystonia itself and some of the clinical symptoms, and what does that tell us about motor control? And then how is that consistent with the imaging findings, and how can we test it further with imaging? So I'm going to jump into this topic of posture. I was talking about movement before, but obviously the postural control system is extremely important when you're moving. So you don't fall over, for example. Obviously when you're walking, your postural system has to allow you to keep your balance. And it's important in sort of stabilizing. If you're going to be writing or something like that, you have to stabilize your shoulder so that you can write neatly. So we know that, and the literature is full of, the motor literature is full of information about both posture and movement itself. There's a lot less literature on posture than there is on movement, though. And the vast majority of postural studies have really been, that literature is very strongly in the animal model domain. But I think it's coming a little more into humans and imaging now. So basically, so there are certain things about posture that are taken for granted. I'm going to argue that there are actually other components of posture that we haven't really thought a lot about that are more directly involved in motor control. And there is a problem in the literature that, especially in the neurology literature, posture tends to be thought of as something static. If you tell someone they have bad posture, you think that they're hunched over, and you think of it as a static thing rather than a dynamic thing. I'm going to argue that we're going to redefine, and we might even want to call it the stabilization system rather than the postural system. But we're going to define posture that basically the brain is using this as a means of controlling and stabilizing the body. And that might happen while you're moving. It might happen while you're still. So this is all. This here is a lot of. It's a lot of words. But I'm going to quickly run through this logic, and you probably won't get it all now. But basically, I'm trying to come to this, describe how I came to this idea that of redefining posture and tying it into dystonia. So I'm just going to take this one by one. So first, basically, the postural system recruits neural programs required for stabilization. So we've just sort of, that is sort of intuitive, and I'm going to sort of define it a little bit more broadly to be all stabilization. So mechanically, if you think about just from a mechanics perspective, the most effective stabilization is going to be implemented by either co-contraction of antagonistic muscles, so you can't move. I was just talking about that before. I was saying, you get this co-contraction of antagonistic muscles. It kind of blocks movement. If you're trying to keep from moving, if you're someone's taking your picture, and you're trying not to move, you'll probably notice, or you might not notice, that you're tensing up a lot of your muscles. And those muscles are all sort of antagonistic to each other and to movement when you're tensing them up. You might use antagonistic muscles, or you might flex a bunch of agonist muscles. Like if you're trying to hold something, if you're trying to keep something dry in the rain, you have a small umbrella or no umbrella, you might use a whole bunch of agonists and pull something really tight up to you to keep it secure. So basically, it's this concept of using co-contraction of bunches of muscles for actually as a functional a way of producing stabilization in the body. So if both of these things are true, then it's possible that is how the body is actually implementing its postural systems. It's actually using these co-contraction mechanisms, which we don't think of it, if you were to sort of overdo it with a co-contraction, it would look weird. It might look like you have a movement disorder. But if you use a more subtle level of it, it may be the way in which the body implements stabilization. So we also know that dystonia manifests as a high level of co-contraction, either of antagonistic muscles or groups of agonists. So if what I just told you here is true, and that is true, then it's possible that dystonia is simply a disorder of amplified postural function. So, I was saying before that basically when you have a disorder, sometimes it can actually show you more about a functional system than you would normally see. It's usually smoothly integrated with other functional systems, but in this case, it's really obvious what the system is. So I sort of mentioned that there are different types of posture, and you would need them in different situations. So there's a consensus already. Nobody would argue with the fact that posture is involved in tonic control of the body's shape. So why is our body like this? Why do we not walk around like that? For example, there has to be some level of tonic control of the muscles to keep our bodies in the position they're supposed to be in. So in dystonias, there are certain types of dystonias called fixed dystonias, where basically the person is stuck in the position. They just cannot get out of it. And in this case, this might actually be a dystonia that is involving excessive activity in the postural subsystem that is controlling resting muscle tone. So basically, that's just they're stuck there. No movement is required for it to happen. So we also know from the postural literature that you need to use posture when in balance when you're walking. So if you're walking, for example, if you did not have your postural systems working, you would fall over. This is not a static system. This is actually a dynamic system, and I'll come back to that in a second. And this is the video I just showed you. Those muscles that are overactive there are muscles that would normally be functioning when you walk, but they shouldn't be functioning that much. So the idea is that in this case, this boy's postural system is working, it's working as it should, but it's working too much. So that in the neurology field is referred to as a movement. It looks like a movement. It looks like he's moving. But I'm arguing that it's a dynamic postural system instead. So it's dynamic postural function. And the easiest way to tell the difference is this doesn't really look like a natural movement. When you see somebody with chorea or with a tic, that looks more like a movement that somebody would actually make. But in the case of this, it's much more, it's sort of jerky. It doesn't look like a motor program. So I would argue they're qualitatively different. So, as I just said, basically, so we already know that, I just said this, hyperkinetic dystonias look like movements. So even if it's stabilizing in the right context and dose, some postural programs are dynamic rather than static. If these two things are true, then hyperkinetic dystonias reflect amplification of dynamic postural programs. So we're sort of moving forward in defining all of these dystonias as postural rather than movements, but there are a few things left. And this is also, this type of system would also be involved in, so in that example I showed you, it's the trunk, but it would also be involved in the neck, because obviously when you stand up, like when you go from lying down to standing up, you have to use these muscles to hold your head up. So if somebody has a cervical dystonia, and there are many different directions the head can go in, it's not necessarily that they're moving their head that way. It may just be that the muscles that are normally all functioning when you stand up are not functioning symmetrically. So what about movement itself? There's one form of dystonia that isn't covered by all of these, if we were to say all dystonia is posture. There's a form, I was saying earlier, writer's cramp is a form of dystonia. There's some dystonias where basically when somebody is performing a motor task, the part of the body that's actually performing the motor task has the dystonia in it. So like if they're trying to write and their hand cramps up like this. Musicians can also develop this. People playing the piano, they're playing like this and the fingers curl under. So how is that posture? In the standard literature, that is defined as two or more competing movements trying to happen at once. How can we argue that it's actually normal movement but abnormally amplified posture at the same time? So there's a lot of literature on how movement and posture are coordinated, and this is thinking of it in the sort of the classic way that when you walk, you need to maintain your posture so you don't fall over, but the walking itself is the motor program. There's no posture involved, in the legs, etc. There's also, Emilio Bizzi has a lot of literature on this, showing, basically arguing that movement and posture are sort of one and the same, and that movement is actually a trajectory of postures. So a third possibility is that movement and posture are actually parallel processes. And I'm going to argue from the neural, so this literature is a lot from the making behavioral and kinematic measurements. I'm going to argue from the neural side that based on the fact that we have these two distinct pathways in the basal ganglia, that perhaps movement and posture are parallel processes that are going on at the same time, and they each play sort of a synergistic and at the same time antagonistic role in controlling movement and not just balance. So an example here is that the movement part of the program would only contract the agonist that is making the movement, whereas the postural program is actually going to recruit both the agonist and the antagonist. So you're going to have a signal from the brain that is, you're going to actually have two signals to this agonist here, but you're also going to be getting activity in something that people tend to think is inhibited. Like when you move this way, you're supposed to inhibit. When you move, use the bicep, you're supposed to inhibit the tricep so it doesn't keep you from moving this way. But I'm going to argue that we actually probably do use the antagonistic muscles, just usually at a really, really low level, and that in such a way that we don't really see that it's there. So the nice thing that this explains is you don't have to do this sort of center surround thing. You have a much more flexible system if you argue that the function of the indirect and direct pathway is not to do center surround, but instead to implement two entirely different sets of programs. And this offers a very nice explanation for why you could have a focal dystonia in the moving body part itself. That it's actually the movement part of the program is just fine, but that the postural program that is actually supposed to be working is overworking. So based on these hypotheses, we can propose, and this is sort of the punchline here, that rather than an inappropriate activation of a movement program, dystonia may reflect an inappropriate level of activation of an appropriate postural program. So that's one of the main things that we're now going to tie back into the imaging. So what this suggests is that we will find abnormalities. So we just talked about the different subtypes of the postural systems, and one of the nice things about this is that you can actually tie it back in to brain imaging and look to see whether you have abnormalities in different subcomponents of the motor circuitry in different types of dystonia. Now, the motor circuitry is really broad, and I'm not going to go into all the different subcomponents now, but suffice it to say that although the basal ganglia is kind of gating what's supposed to happen, it's actually the downstream regions that probably have the programs coded in them. So, for example, it may tell the cerebellum that it's supposed to do one thing, and the cortex is supposed to do something else. And in different forms of dystonia, it may be that in one case, the balance-related motor function is not working properly, and that might, for example, you would see that in the cerebellum. Whereas in another case, it is the control of movement itself that's not functioning. You most likely see that in the primary motor cortex. So this is going back to some old data, but also bringing in some new data to compare two different types of dystonia. These are some findings that we had, and basically, just to summarize, this is an fMRI time course. The task is finger tapping, and it was done in patients with focal hand dystonia, and down here, more recently, cervical dystonia, and compare with controls. And basically, we found that in the putamen, so this one of our motor structures of great interest here, you see in the patients, you actually see that they're sort of ramping up each block of tapping. It's getting higher and higher, and the function in this region is not coming back down to baseline. And in the controls, it does. I mean, normally in a block design, this is what you see. More recently in cervical dystonia, we see something very similar. The timing is a little different here. This has 30-second rest blocks in between, so they're just resting their hands. And this one actually has a minute, so that's why the time looks a little different. In addition, the amplitude in the next slide, at least, is going to look a little different because this is actually bilateral finger tapping, so this is both hands, and this is unilateral. But we get the same result either way. So in the basal ganglia, it looks the same in these two different types of dystonia. But in the cerebellum, it doesn't. And so in this case, in the focal hand dystonia patients, they pretty much, this isn't 100%, but they pretty much come back down to baseline in the cerebellum when they're doing this finger tapping task, same task. And in the cervical dystonia patients, it actually looks even worse than it looks in the putamen. So this is exactly what we would have predicted, because we predict that in the cervical dystonias, it's more of a balance-related postural circuitry, whereas here it's going to be more the motor control circuitry. And I didn't put this in the slide, but so you would predict here that you might see this in the primary motor cortex, or see the elevated baseline or at least increased function in the primary motor. You actually don't see the elevated baseline, but you see a huge difference in the amplitude of the primary motor cortex in the focal hand dystonia patients versus their controls. And in the cervical dystonia patients, you see no difference. The time courses look almost identical in the amplitude across the patients and controls. So this is a really fun way that imaging can test these hypotheses, and obviously there's a lot more that we're working on analyzing right now. So yeah, this is a very specific way that we can look at sort of the subcomponents of posture and their potential role in dystonia. So now I'm going to tie this back into those collateralizing fibers. So we just talked about all these different types of postural subsystems. And I talked about the cerebellum, primary motor cortex. How do you coordinate all this? At any given time, your body has to know, should I be in rest mode? Should I be in movement mode? Should I be, how can I control what my hand is doing while I'm writing, while at the same time controlling what's going on with it, making sure my shoulder is stable enough to write. So the really nice thing is that these collateralizing fibers that go to all these different motor regions suggest a perfect way to coordinate all the different subcomponents of posture. And that's the real punchline, is that perhaps we have found maybe what the function of these fibers is. Clearly, when you have something that's projecting that extensively, it is doing something to coordinate multiple motor systems. So this is a simplified diagram here. It doesn't include the projection to the red nucleus that we know is there. I'm just for purposes of simplifying this. This here is a circuitry diagram where we're mapping out a theoretical circuitry. Well, no, it's not, most of it is not theoretical, most of it is known. We know that these fibers, there are these simpler fibers that go to the thalamus. They also go to the PPN, but they don't go to the red nucleus. And then there are the more extensively collateralizing fibers, which are represented here by the ones that go to the PPN. And so the idea is that these are the two parallel systems. This one's controlling movement or gating movement, and this one is gating the postural programs. And the great thing about these collaterals is with all you need is one neuron, and you can send the same signal out to all these different regions. It's an extremely efficient way to set up the system. And furthermore, now this is the only part here that's theoretical. We know there are inhibitory interneurons in these, every region of the brain pretty much has inhibitory interneurons. If you were to argue that the PPN projection here actually synapsed on either an inhibitory interneuron or an inhibitory neuron that projects to a region further down, that you, with this single signal, can, say, activate a program that is stored in the PPN or further down in the brainstem, and at the same time inhibit the program up here. So if I was saying the body has to know whether it's supposed to be turning on the movement-related or the rest-related programs. I'm suggesting that this is some sort of toggle between, that these collateralizing fibers can set up a toggle between the two systems. So the brain can never get confused which system is supposed to be on. When this program is turned on, this one gets inhibited and vice versa. So we haven't proven this yet, but I think the anatomy can give us a lot of clues as to what the function is, how the functional system is set up. And so obviously now, going forward, we're going to try to work more on dissociating these two functional subsystems and on figuring out whether the circuitry is what it's really doing. So how does this model address the needs of a motor control model? So I'm going to basically just go through all the points that I went through before and say specifically how this model addresses them. So the first thing that we wanted to improve was to include a factor that allows for qualitative as well as quantitative differences between movement disorder symptoms. And I knew I wasn't going to have a lot of time to go into other movement disorders, but this is the point at which I will mention a few things about that. So if you have two functional subsystems, each subsystem is going to look qualitatively different. You have a motor system and a postural system. In addition, you actually have multiple postural subsystems. So you have all these different functions of the circuitry it's no wonder you can get such a broad range of symptoms in movement disorders. You can have something that looks more like a movement, something that looks more like posture, or something that looks somewhere in between. For example, in Parkinson's disease is classically thought of as something where basically there isn't enough signal to move, so they don't have enough of a stimulation of the basal ganglia circuitry to move in the first place. And that is actually true. So if we go back to this here, this actually is a good model of Parkinson's here. The net result of the cell loss in the area that's substantia nigra is what's going on here. You actually get increased output from this region, which inhibits the thalamus and motor cortex. So this is what you would get. But I'll argue that it's actually two things happening at once. It's a double whammy, I refer to it. As in addition to having trouble activating the movement programs, you're getting a massive output to the resting postural program. So that, I'm arguing, is where it results in the bradykinesia and rigidity you get in Parkinson's, where basically the baseline muscle tone is really, really high and they're really stiff and they have trouble moving. So not only are they having trouble activating the movement signal, but they're getting a big mechanical block by overactivation of the resting postural signal. So that's an example of how this can apply to other movement disorders and how it can actually make it much easier to explain some of these disorders and some of the differences between them. So I was also referring to the possibility of assigning more active rather than passive role to the indirect pathway. And I would say we've done that here in assigning actually a completely, not a new role, and we know the basal ganglia are involved in posture, but by saying that it doesn't just sort of act as a redundant inhibitor to the direct pathway. The challenges of the center surround concept in the sort of mainstream model. Basically with this model we no longer need center surround. This idea that you're actively inhibiting all your other motor programs every time you move just isn't very efficient. There is, of course, there's tonic inhibition of these programs there anyway, but it doesn't have to be implemented in a center surround mechanism. And basically the nice thing about this is that it allows us to assign an active role to the indirect pathway. All the circuitry is not simply there to kind of assist the direct pathway. But the great thing is that it's still consistent with experimental observations. And one of the most interesting things I've always found is that since it's thought to inhibit movement, the idea that that has always been sort of a central inhibition of movement, at least in my understanding. Here we're arguing it really appears as more of a mechanical inhibition of movement. It's a cost-benefit ratio of the level of stabilization relative to the fluidity of movement. So there's always going to be a balance between those two. So, whoops, sorry, I went back there. So finally, create a model that allows for the heterogeneity of etiology across movement disorders. And as I said before, it allows for multiple hits leading to similar outcomes because it's a systems level model. And it also shows how you can get dystonia, for example, both when dopamine levels are too high and too low. So I'm going to quickly run back to this one again. So I remember I mentioned in Parkinson's when their medication is at its peak and at the end you can get dystonic symptoms. And you can argue from this that if you have too much or too little dopamine, which is what is driving the function of this neuron here ultimately, if you get overamplification of this output program or this output program, in either case, you could actually get dystonia. So just to summarize sort of the model in general, so basically we're arguing that all dystonias may reflect abnormal amplification of brain postural function. The brain postural system may be a critical modulator of motor control itself. The existing imaging data that we have, as well as a lot of other data out there in the literature, suggests potential anatomical and physiological mechanisms that implement the proposed model, including the circuitry in these collateralizing fibers I mentioned. And the model has implications both for previously unexplained and sort of puzzling symptoms that you see in dystonia, and also for the qualitative range of symptoms across the different movement disorders. So that is it. And these are for the movement disorders. I have quite a few people that I've worked with in the past, and I also work with a bunch of people in mood disorders and addiction as well. So I would like to thank all of them, especially for all their hard work and data analysis.